So now we come to the business of tonight's meeting, which the title of our talk that we'll hear tonight is Evolution in the Genome, Studying Brightly Colored Tropical Butterflies to Understand Evolution and Speciation. Now I'm eventually going to hand over to my esteemed Vice President, Blanca Huertas, here to introduce the speaker, but I just want to tell you one thing about him before we start. Chris was a graduate student with a very good friend of mine, Jim Mallet, and I remember him, as a graduate student, teaching my son, who was about 3 at the time, to chase morpho butterflies down a muddy road at which my son would fall over from time to time. And all kudos to Chris is he laughed, which was absolutely excellent. So Blanca, over to you.
Hi, good evening. We're going to introduce Professor Chris Jiggins, who don't need much presentations. He's one of the authorities on the butterfly world, and he is currently the Director of Studies in Natural Sciences at St. John's College in Cambridge, and a Professor of Evolutionary Biology there. His research interests include the study of adaptation and speciation of the Lepidoptera, which includes the butterflies and the moths. And in particular, his topic is how species converge due to mimicry as a model for understanding the predictability of evolution and the genetics and ecological causes of speciation. Professor Jiggins also will talk. His talk will cover how wing pattern signatures in the genome are exchanged between species through hybridization and recombined to generate novel combinations, highlighting that the patterns of sharing among species and challenging our understanding of species as units of evolution. So we're going to welcome Professor Jiggins, and thank you very much for coming to the Linnean Society.
So hopefully you can hear me. Thank you very much for coming. And it's great to be here under the rather august glare of Wallace and Darwin up there. It's a little bit intimidating. So I'm going to talk about our work on these South American butterflies. So I'm going to start, if I can get this to work. Here we go. Oh, no, I need to turn it on. I have to remember that. Okay, I'm going to start by taking you to Colombia in honor of Blanca here. So a couple of years ago, we flew down to a place called Mocoa in the eastern Andes of Colombia in search of butterflies. And this was a very exciting trip. My PhD advisor, Jim Mallet, who's already been mentioned, had been there in the 1980s, but it was a part of the world that was very dangerous to visit for a very long time, so we were unable to get there. This is me waving a red rag, which attracts these red-colored butterflies, and you can catch them. And so we were out there in Colombia in search of a particular butterfly, which I'll show you in a minute. But it was an exciting trip because of the peace deal in Colombia had just been signed. And this part of the world that had been extremely dangerous, suddenly was accessible. And this is sort of an exciting time in Colombian history, really, when large parts of the country that were very dangerous were, suddenly, relatively more peaceful. And so what were we looking for? Well, here's us catching butterflies. So sometimes they fly and feed on flowers very high up, so a long pole is quite handy. And here are my Colombian collaborators, Carolina in particular here, and Camilo, who I've been working with for many years. What were we looking for? Well, in particular, we were looking for this butterfly, which was described back in 1996 by Andrew Brower. He called it Heliconius tristero. And it really hadn't been found since that, I think, apart from 2 specimens caught by Blanca somewhere in between the sort of 20 years. 1998, there we go. Well, it hadn't been found since 1998, which was quite a long time. And so we went in about 2016 to look for these things. And we went to the town of Mocoa, and we went up into the mountains above Mocoa, and almost immediately found lots of them. So there was no problem finding them. They were there. They just hadn't been a part of the world that was easy to find. So why was this interesting? Well, it's sort of part of a story of discovery over the last 15 years of a lineage of Heliconius. So if you'd asked me when I was doing my PhD, are there species of Heliconius that we don't know about? I would have said, well, we know the taxonomy really well, and this is one of the best studied groups of tropical butterflies. And there probably aren't going to be many new species. But since then, we've found this lineage of butterflies that was hidden, and I'll explain why it was hidden in a sec. So there were two species known. This is Heliconius timoretta from eastern Ecuador, Heliconius hirripa from eastern Colombia. And then the first one that was discovered, a new one that was discovered, was this thing Tristero by Andy Brower. But after that, we also found a whole range of populations down the Andes over the coming years. And here are the years and the names of the people that describe these populations. Now, why were these big, colorful butterflies so hidden? Well, the reason was they are perfect mimics of another species, Heliconius melpomene. So anyone who had caught them just thought they were Heliconius melpomene, this very common, widespread species. And it was only once we started sequencing their genomes that we realized that they were actually a distinct cryptic species there hidden in these populations. So that was quite exciting. And we actually now consider all of these things as to be geographic races or subspecies of this one, Heliconius timoretta. Apart from this one, actually, which we still call a species for whatever reason. Anyway, so that was the reason for our trip. And so now I'm going to just sort of give you a little bit of a tour of the biology of these butterflies, because they do some unusual things. And the first thing they do that's quite interesting, I'm just going to make sure I've got the time here, is they feed on pollen. So they very systematically visit flowers and collect pollen on their proboscis. You can see this sort of glob of pollen here. And then they go and sit on a leaf and sort of excrete liquids onto the pollen and process it, and then suck up the liquids. And from that, they get a very reliable source of amino acids. So generally, butterflies or Lepidoptera as adults primarily feed on nectar, and so mainly are feeding on carbohydrates. But these guys have a very reliable source of protein and nitrogen, and that gives them the ability to live for a very long time. So they can live up to 8 months in the wild as adults and be continuously reproductive throughout that time. Some butterflies live a long time, like over the winter in the UK, but they're not sort of active and flying and reproducing for all of that time. So that gives them a quite unusual biology. You might notice they have quite large heads. They actually have very large brains relative to other butterflies. A friend of mine who studies brain evolution in these things has compared the rate of brain increase relative to body size and says it's similar to that in the human lineage. So it's a model system for understanding brain evolution. There we go. And they learn their way around the forest and find the resources that they're going to visit. And one of the things they're looking for, the females in particular, what did I do? Oh, there we go. I pushed the wrong button. Are these Passiflora vines. And so these are passion fruit vines, and they're packed full of cyanogenic compounds. So these are toxins which the butterflies sequester into their bodies, and that makes them nasty to eat for predators. And that's why they're so brightly colored, is that they're warning predators that they're bad to eat. They also have some slightly unsavory mating habits. So this is a species called Heliconius charitonia, which was photographed in the garden of Andrei Surikov in Florida. And what you can see here is some males, and hidden under this males is a pupa of a female. And you can see here, this is the female pupa, and these males have stuck their abdomen into this pupa and are mating with the female. These are two males here. The female is still inside the pupa, and they're basically mating with her before she comes out of the pupa. It's a very sort of unsavory, really. So the poor female, there's those two males again, and here's the poor female just sort of emerging into the world already kind of mated by this male. So the female has no choice in terms of who she mates with in this case. There are other species that are a little bit more civilized and mate normally as adults. This is Heliconius melpomene. They also, another sort of curious behavior they have is that they sleep in little groups or roosts, and they spend inordinate amounts of time sort of, sorry, I'm just getting used to this thing, fluttering around each other in the evening. So from about 4 o'clock in the evening in the rainforest, little tendrils and vines in rivers, you start to find these aggregations of the butterflies. And they spend enormous amounts of time sort of fluttering around each other. It's a bit like kind of, negotiating with your spouse the best spot in the bed. And then they all hang up and sleep together. And we don't really know why they do it. The males and females sort of rather randomly flutter around each other. So it's nothing to do with courtship as far as we can see. But it's sort of interesting social behavior. Okay. But the main reason we study them is because of their amazing diversity of wing patterns, and you can see just a sum of that here. So these are different species, different groups of species in the rows. And so you can see, for example, here within Heliconius melpomene, you've got two very different forms in different parts of their geographic range. And then the very closely related species, Sydno, looks very different. So you've got closely related things that have very different wing patterns. But then also you've got this mimicry, sorry, I keep pushing the wrong button, where you've got all these different species and even different genera up here that have all converged on the same pattern. So you've got great diversity, but also convergence and mimicry. So why are they mimicking each other? Why are they looking similar? Well, it's sending a signal to predators that they're bad to eat. And so this was originally recognized by this amazing naturalist, Fritz Müller, who was a German naturalist who lived in Brazil, a frequent correspondent with Darwin. And he recognized that there's a benefit to toxic species to look similar, and that's Müllerian mimicry. And here you have different forms of Heliconius melpomene along the top and Heliconius aurato along the bottom. Wherever the two live together, they look identical. But as you go across their geographic range, they look very different. So just within Ecuador, East Ecuador, in the valley, the Puyo Valley, they look like this. In the Amazon, they look like this. And on the west in Ecuador, they look like this. So in a very small space of geographic space, you get these very different patterns in different places. They look so different, you would never think they were the same species, but we know genetically and from the fact that they interbreed freely with each other, that they really are the same species. So this great diversity, this sort of flowering of diversity among very closely related forms is a great in to try and understand the genetic basis for diversity of patterns. Because this means we can cross together different things that have different patterns and do genetics like Mendel did. And so we've done that over many years. And this is a slide now that summarizes, really 15 years' work, where we've actually now identified the specific genes and how they work that control this diversity of wing patterns. And the amazing thing, actually, is that this enormous diversity of wing patterns that we see is in large part controlled by a very small number of genetic loci, a small number of genes. And just to sort of summarize these quickly, there's one called Wnt, which is a signaling molecule. It's a molecule released from cells that diffuses and signals between cells, called Wnt. There's a gene called Optix, which is a transcription factor. So that's a protein that binds to DNA and controls the expression of other genes. This one actually, it's a question mark, but it's actually published now. It's another transcription factor called Aristolus 2. And this is a one which is rather puzzling, which we call Cortex, and we don't really know how it works or how it acts. But it's actually the same gene that the famous peppered moth, who some of you might have heard of, that evolved to be black in the Industrial Revolution, the same gene that made the peppered moth black is the one that makes these yellow and white stripes on these Heliconius butterflies. Okay, so I'm going to tell you a bit more about what these genes do. So Optix is perhaps the best studied, and this is a stain showing in pink the expression of Optix in the wing of this butterfly here. And what you can see is that essentially wherever you're going to get a red patch in the adult wing, you get this expression of Optix. And so Optix is sort of like a paintbrush that paints red onto the wing, and wherever you turn on Optix, you get a red patch in the wing. It's like a sort of paintbrush painting colors onto the wing. These are actually, what you're seeing here is the wing from a pupa. So this is, although the shape and the form of the wing is very much like the adult, it's actually tiny. It's from the pupa before the butterflies emerged. And this up here is an experiment where if you knock out, in other words, you break Optix in this butterfly, you can see here's the normal butterfly, and here's one where Optix is not functioning, and you can see you lose patches of red. And this amazing experiment here is Agraulis vanilli, where all of the orange gets lost. This is the normal one, and this is the one where it's been lost on the right here. So I'm going to tell you a bit more about those experiments and how they work now. So what we're doing is we're using this technology called CRISPR, which many of you may have heard of. People are very excited about it as a technique in medical genetics and generally for manipulating genomes. And what it is there's a protein from a bacteria which targets DNA and cuts DNA into sort of, makes what's called a double-strand break in the DNA. But the amazing thing about CRISPR is you can put in another molecule, an RNA molecule, and you can target that cutting to any specific location in the genome. And that's the sort of really exciting thing about this technology. So you can really target your break in the DNA to a specific place. I'm just going to have a glass of water. So what we're doing is injecting into an egg, a butterfly egg. Randomly, some of the nuclei in the egg will have induced these double-stranded breaks. And then the cell will sort of repair these breaks, but it tends to make mistakes. So it kind of sticks the DNA back together, but makes a mistake and adds a bit or removes a bit or puts in a different base. And so if we do this cutting in the coding sequence, the protein coding sequence of a gene, we tend to induce mutations that break the gene and stop it functioning. So we're asking, like, if it's like trying to figure out what different parts of a car do by hitting them randomly with a hammer. It's quite a crude experiment, but as you'll see, the effects are quite dramatic. So the nice thing, of course, is that we can then, just in the first generation, we can see these butterflies come out and we can see what their wing patterns look like. And here's an experiment where we've done this for this Wnt gene in this butterfly. This is what it looks like normally, Heliconius sera. And here are some of the ones with these edits. And what you can see is some of them are quite mosaic. So in this one here, for example, or here, some of the cells have got the gene working. And that's where they look normal. And some of the cells, the gene is not working, and they've turned yellow. But essentially what this gene does is it patterns this part of the wing and makes it black. So when you break it, then the wing goes yellow. So this tells us something about where in the wing this molecule is patterning the wing pattern. And here is some more of those experiments in different species, and you can see there's quite a diversity of effects. So in particular, I'll point out here, we've got the wild type on the normal butterfly on the left and the knockout, the experiment on the right. You can see in this erato, it sort of makes this big expansion of red. But if you look at the mimic of Melpomene over here, it just makes a little yellow patch here. So the effect is quite different. The same mutation, its effect is quite different in different butterflies. And I'm sure I ought to be able to telling you something very intelligent about what this means, but I think we don't really know quite what this means. But there's something different about the genetic background of how you make a pattern in these different species that means that we get very different effects of these knockouts. For example, here, this one, you always get this yellow patch in the very tip of the wing that's not there normally, whereas this one, you never get yellow in the tip of the wing. So there's something different happening. So it's starting to tell us something about how it's not just the diversity is coming about by the sort of evolution of the whole genetic system that makes the wings. But I'm not, I guess, as I say, we're not really quite sure how to interpret these patterns yet. If you look at these are now the same experiment, but within different forms of the same species. These are all much more closely related to each other than the previous slide. And you tend to get the same effect. So it always affects this same patch of the wing. It either turns it red or yellow, depending on what you have there in the wild type butterfly. So in this species, this Wnt gene patterns this little patch of the forewing here. Anyway, these experiments are quite fun. It's kind of like playing God with butterflies. And then this is the Cortex gene, the one that makes the peppered moth black. And this does something completely different. It just turns black scales to yellow scales. And it seems to have no kind of spatially localized effects. We just get these little patches sort of rather randomly across the whole wing. So in this case, the breaking the gene is not really telling us anything about the spatial localization of the patterning. We think this gene is probably important in making scales everywhere. And we don't really quite know, why it's sort of, in the genetics, it's specifying this yellow band. But it seems to be expressed everywhere. So we think there's something going on that we don't understand there as well. OK. So now I'm going to change tack a little bit and talk a little bit about the history of these genes. So now we understand where the bits of the genome that are important for controlling the diversity of patterns, we can ask questions about where the variation comes from to make all this diversity of patterns. Okay. And so, we've been interested in speciation, the process of speciation in my lab for a long time. And this is sort of the idea of the branching of one lineage into two, to make, let's say, two different populations of butterflies with different patterns. But occasionally, because this is a gradual process, species will produce hybrids. And so sometimes, if the barriers between them are incomplete, hybrids will be produced, and the hybrids offer an opportunity for genetic material to move between species. And so if that hybrid now mates with one of the parental types, it can produce an individual with a genome that's a mixture of the two species, and as these continue to mate, they provide a route for genetic material to flow between species. And so we can think of the genomes of the two sort of species here as actually being somewhat porous with the potential for mixing between these genomes. And of course, there's something that keeping these things as different species. We're still calling them different species, so there's something making them different. So what we think is that some regions of the genome will be resistant to this mixing, whereas other regions might be more free to mixing. Now, so typically people think of hybrids and hybridization as a process of kind of that removes the differences, a sort of destructive process of kind of removing the differences between species. But we're particularly interested in the possibility that actually hybrids could provide a route for acquiring useful genetic variation. And so, if we think about new patterns arising in evolution, we typically think of that, sort of waiting for new mutations. You have to wait for a random change in the genome, and sometimes that will be beneficial. But actually, hybridization could introduce genetic material into a species that's sort of already been tested by evolution in another related species nearby, and actually might provide a more efficient source of genetic material for adaptation. And as we kind of sequence genomes of lots of different species, we're realizing this is an important process. And so here is a paper that studied the Lake Victoria cichlid radiation. And what they showed is that genomes of this sort of amazing radiation of fish in Lake Victoria actually came from a mixture of Congolese basin and Upper Nile basin fish. So actually, at the base of this huge radiation of hundreds of cichlid fish, there was a mixing event of two very divergent lineages. And what they argued in this paper is they think that this generated a lot of genetic variation that then allowed the species to radiate. Another sort of classic example are these Darwin's finches, where this is a gene, this is the evolutionary history of a gene that controls whether they have blunt or pointy beaks. And the history of this gene looks very different to that of the species. And in fact, Geospiza fortis, I can't remember which species that is here in English, but Geospiza fortis here has some individuals have the pointy allele, and some up here have the blunt allele of this gene, even though these alleles are extremely ancient, relatively, much older than most of the radiation. So these ancient alleles are being sort of shared around between the species to make kind of, whether your selection is favoring pointy beaks or blunt beaks in that particular population, you can sort of make use of this ancient variation that's sort of floating around in the populations. And this is another example, which I quite like. It's a big radiation of these birds called munias, which have very different coloration. And there's a whole load of different genes that are shuffled into different combinations to make these different patterns. I'm not really going to go into that. So we think the butterflies are doing the same thing. And in particular, I'm going to concentrate on this, the species that we've talked about, so Melpomene and its sister Heliconius sydno. They live together in the Andes, Western Andes and Central America. And then Timoretta, which is found down the east of the Andes, which I already mentioned. And so we know that hybrids between this sort of Melpomene lineage and the Timoretta lineage are found, but they're very rare. I've never found a very convincing, I've caught thousands of these things in my life. I've never found a really convincing hybrid, but there's sort of 40 or 50 in collections around the world. So they do hybridize, but very, rarely on a sort of per individual basis. So it's not that, they are really good, and to persuade you they really are good species, before I go on to tell you how mixed up they are. So, what we found was that these populations of Timoretta, the ones down the east of the Andes that look just like Melpomene, and that we sort of hadn't found for all those years because they looked so similar to this other species, they actually look so similar because they've stolen each other's genes. They've actually, the genes that make the patterns actually are shared between these species. And so this shows a cartoon up here of the genome here. This is the optics gene, which controls the red patches. And there's a region here, which is these are genetic switches that turn optics on and off in different places in the wing in this region here. So if you look at most of the genome, you find that populations of Melpomene and populations of Timoretta cluster with each other, as you'd expect, even though they look quite differently. And so this kind of pattern that you find here with these two clustering together, this is from Colombia and this one from Peru, and then here again, Colombia and Peru. But the species cluster with their own species across most of the genome. But then when you look in this region that actually controls the red patches, suddenly these things flip around and they start to cluster by their appearance. And so actually the two Colombian ones look very similar, and the two Peruvian ones look very similar. And then as you sort of move along the genome again, you go back to the normal species clustering on either side of this region. So there's a very narrow little region of the genome where suddenly the genetic variation recapitulates not the species relationships, but actually just the appearance of the butterflies. And so what we think is this is a signal of genetic material moving between these species through hybridization. And so actually, if you want to mimic another species, the easiest way to do that is to just steal their genes and kind of evolve a similar pattern to what they look like. So that shows that we think that hybridization and movement of genes between species can facilitate mimicry and the evolution of similarity between different populations. But it can also generate novelty, and this is another butterfly that Blanca's quite familiar with. So this is Heliconius hirripa from eastern Colombia. You just need to drive down the Andes from Bogota towards the Llanos, and you find these things flying on the slopes of the Andes. And for a long time, since the 1970s, it was sort of proposed that this thing might be a hybrid, because this rather typical sort of pattern of red and yellow here is only otherwise seen in hybrids. But this is a really good species. It's kind of this consistent. You find big populations of these things, they all look the same. But Camilo Salazar and his colleagues in Colombia showed that actually you could make a hirripa by crossing these two species, Melpomene, and Cydnocordula. So if you cross these two together, it's sort of visually rather appealing, isn't it? Because this one's got a yellow stripe and this one's got a red stripe, and you put them together and you get the double stripe. And actually then when we sequenced the genome, we showed that that's true, that the red gene of hirripa looks like Melpomene, and the yellow gene, which is Cortex, looks like Cydno. So actually this is a novel pattern which has arisen in evolution through hybridization and mixing of different genomes. Okay. So the red, in this case, the red and the yellow are controlled by genes on different chromosomes. They're very distant, they're not linked to each other. But actually, this mixing can happen on a much finer scale, and I'm going to explain that in a sec. But first, I just want to tell you some rather strange history. So this is what we call Dennis. This patch of red on the wing is called Dennis, for obvious reasons. It's named after Dennis the Menace. So Dennis the Menace, this is an amazing story of convergence, because Dennis the Menace, there's an American Dennis the Menace shown here on the left, and a British Dennis the Menace shown on the right. They were both published in the same month in, I think, 1953, completely independently on either side of the Atlantic. And I think they're both in continuous publication ever since, as far as I'm aware. So anyway, convergent evolution. And unfortunately, I think the patch is named after the rather less menacing American Dennis the Menace. But anyway, there was a butterfly in Trinidad that had a very odd pattern, and Jocelyn Crane and William Beebe named it Dennis the Menace. So we're interested in Dennis. And actually, just Dennis and these rays here. So these are two red patches on the wing, which generally are found together. And we sequenced the genomes of lots of butterflies. And we showed that if you have both red patches, you have this kind of red pattern of variation. And if you have neither of them, you have the sort of blue pattern. But there are butterflies, there are populations out there that have one or the other. And just show that. So yeah, so here we go. If you have both, you have the sort of red genetic variation. If you have neither, you have the blue genetic variation. But if you have just Dennis, then you have this. You always have this this red patch here. Sorry, no, yeah, this one, this red patch here. And if you have rays, then you always have this red patch here. So there's two little bits of the genome. This one makes the rays. This one makes the Dennis. And this is just a few thousand bases of variation. This is a tiny, tiny, there's 270 million bases in the butterfly genome. And we're just talking about a few thousand of them here. So this is a tiny, tiny proportion of the whole genome. But when we look across the species, what we find is that across very different species, we find the appearance of these sort of little bits of DNA that control these different patches. So up here, you can have Dennis and rays, or just Dennis, and down here you can have Dennis and rays, or just rays. So these same little bits of the same little genetic switches appear again and again in these different species. And when we looked at the evolutionary history of these bits of the DNA, what was really surprising was they look very different. So even though these are right next to each other in the genome, they have very different evolutionary histories. So we think that the Dennis controlling bit originated in this radiation of butterflies, which we call the sylvaniforms. And then the ray bit originated in this group, which is the Melpomene group. And you can see these two groups of butterflies diverged from each other about 4 million years ago. So they're really quite genetically different. And so what, no, didn't want to show him yet. And so what we think is happening is that these little bits of DNA are switching between these species through hybridization. And it's happening repeatedly in order to, you have to hypothesize lots of events in order to get all of these, these very similar genetic switches into all these different species in different combinations. So there's this process of exchange of DNA between these lineages, these species, is happening quite frequently. And and I gave the game away. So what's exciting is that this is also true in our own species. And so since they sequenced the Neanderthal genome a few years ago, we've known that bits of the European and out of African human genome also contains little pieces of Neanderthal variation, which we think has come about through hybridization between humans and Neanderthals. And I got my genome sequenced, well, not my whole genome, but you spit in a jar and you send it off to 23andMe for $100, and they send you back a login to their website, and it tells you all sorts of things about yourself. But I am 2.8% Neanderthal, which was at the top end of their Neanderthal variation, which I was very proud of. So anyway, so the same kind of analysis methods that they used and developed for the Neanderthals are what we've been using to try and understand these butterfly genomes. And this genome sequencing has sort of added, I think, the sort of the traditional view of species as sort of distinct evolutionary units that kind of go off on their own trajectory. It's actually much more complicated than that, and much more exciting, I think, in that useful genetic variation can be exchanged between species, and parts of the genome might be quite mixed, and other parts might be quite different. It's much more complex and heterogeneous than people have thought in the past. Okay, so one more little story, and I think I've just got time for this. So we're also interested in other things apart from color pattern, and in particular, what about behaviors? What about the genetic basis of behavior? So these Heliconius, this is a Heliconius cydno, rather futile courtship attempt here. They go on for hours doing this, and often nothing happens. But the males use the colors to find females. So they the red butterflies prefer to court red females, and we think that's sort of really the way to find a female is to sort of look out for the right color pattern. So if you're a Melpomene, you prefer to court red females, and if you're a Cydno, you prefer to court white or yellow females. And so these species, kept distinct by this because they prefer to mate with their own type. But in order to evolve this system, you need a change in the color of the wing, but you also need a change in the behavior. And we don't really know how that's encoded, whether it's in the visual system or in the brain or anything like that. We don't really know, and no idea. But we're interested in the genetic basis for that behavior. So can we find out anything about how many genes are there that control this genetic change in behavior? So in order to do that, we did the same sort of thing again. We did crosses and sort of did traditional genetic techniques. So the grandmother here is a black and white Heliconius Cydno, and we cross it with a Heliconius red and black Heliconius Melpomene. Now, these emphasize that these are really good species. The F1 females are completely sterile, so we can't do anything with those, but the males are fertile, and we can cross these males back either to Heliconius Cydno here or to Melpomene. Now, if we cross them to Melpomene, all of the offspring have a strong preference for red. So the genes that make you like red are quite dominant in this cross. So that's not very useful. So there's not much variation there. But if we look in the back cross to Cydno, there's lots of variation such that some individuals prefer red and some individuals prefer yellow, or prefer white. And so my then PhD student, this is sort of to give you an idea of the amount of time this experiment has taken, my then PhD student, Richard Merrill, and he now has his own lab in Munich in Germany. So from when we started gathering these data during his PhD, and we published the paper actually in this January. So it's been at least 10 years of kind of collecting doing these crosses. And the reason is it's really hard to get measure the behavior for lots and lots of individuals in order to do the genetics. We need to know the mating preference of all of the hybrids that we produce in order to then correlate that with the genetic variation and find out which regions of the genome are controlling the difference in behavior between the butterflies. And that's very laborious. So, but it's sort of quite, it's quite exciting because you spend years and years doing these experiments with the behavior, and then you sequence the genomes of all these hybrids, and then you kind of bring all of that together, enormous amount of information, and you push a button and see what happens. And so what Richard found was actually remarkably sort of similar in some ways to the wing patterns in that this behavior has a relatively simple genetic basis. So he found 3 regions of the genome, 1, 2, 3. So these are the 21 chromosomes along the bottom. And the little plots show the likelihood score, the probability of association with the phenotype. So there's a region of the genome on chromosome 1, another one on chromosome 17, and another one on chromosome 18 that have a strong effect on this behavior. So it's actually not that complex. You would have thought maybe behavior would be a lot more complicated genetically than patterns on a wing. But actually, it's sort of somewhat similar in a small number of regions of the genome control a lot of the differences between these species. And what's even more cool is that actually chromosome 18 here, and this end of chromosome 18, is exactly where the optics gene is that makes the red patch on the wing. So actually the gene controlling the preference for red and the actual gene making the wing red are located in the same bit of the genome. And we think that should be, that's sort of a genetic architecture that should make speciation easier, because if you have these genes linked, it's harder for them to get broken up by recombination. And so, so what was it? Oh yeah, and so it's sort of interesting to think, well, could this process of this process that I talked about in the wing patterns, where you sort of put together new combinations of genetic variants to make new patterns, could that actually happen for a behavior? So could you put different genetic variants into new combinations and make butterflies behave in a different way? And so there's some quite sort of tentative evidence, and I don't think it's very convincing just yet, but this was a paper that we published quite some years ago, actually. A master's student in Mauricio's lab in Bogota, Maria Clara Melo. She generated these Eurypa-like hybrids. So in other words, she crossed these things together and made hybrids that looked like this. So if you wave little paper models at these butterflies, the yellow ones prefer yellow, yellow wings. And this is what I spent my postdoc doing, was cutting out little paper butterflies, and we stuck them on the end of a barbecue poker and sort of waved them at the males. It's a very high-tech experiment. And so if these ones prefer yellow models, and these ones prefer red models. But actually these guys, these hybrids that were made in the lab, preferred a model that had both red and yellow. So in other words, by making a hybrid between these guys, you could actually make a genotype that had a different genetic preference. So we haven't done any genetics on these hybrids. This was done long before we kind of knew all of this stuff. But it's sort of suggestive that maybe the behavior could evolve in the same way as the color patterns by sort of shuffling variants into new combinations. Okay, and so I think I should probably just about finish there. So just to summarize, I think there's this emerging story from these genome studies of lots of different diverse radiations, sort of diverse systems of birds and fish and butterflies, that often the genetic variation that controls the novelty is actually much older than the species that we're looking at. And a lot of the novelty comes about by shuffling ancient variants into new combinations. And that's sort of quite exciting. I think it's quite a different view of evolution to maybe what we would have thought before, where we sort of thought that you had to kind of wait for new mutations to come along before you could evolve new phenotypes. Maybe behavior might evolve the same way. I think that's a bit speculative. But this mixing between genomes can provide the starting material for adaptation. But they're genetic variants that have already been tested by evolution. And so finally, just to thank many people in my lab over the years who've helped. And really, this slide is an excuse for me to plug my book. So if you're interested in Heliconius butterflies, what better way to buy your friends a present than buy my book? Okay, thank you very much.